up there. So to start, I'm Ellen, and I'm a neuroscientist studying the networks in the brain as they develop. So you might be wondering then why I'm showing you these two images and what they have in common. So on the left, we have a zoomed in view of the image that's currently on display in the Koch Institute galleries. And this shows a cross section of a brain that was in the process of growing new neurons during normal brain development. The question that I want to ask of each of these new neurons is the same as what so many people are asking when they purchase one of these kits, namely, who am I, who are my ancestors, and where did they come from? So rather than constructing a family tree of people, my work seeks to build a family tree of neurons. And I do this using a type of DNA barcode, which not only allows me to generate cells with this range of beautiful colors, but also allows me to ask about the family relationship between the roots of the family tree, these neural stem cells down near the bottom of the screen, and the daughters or offspring that are migrating out to find their final positions in the brain. And this is much in the same way that I might want to trace back Italian heritage to ancestors like my own, starting from asking my family members after they had migrated to North America. So in the developing brain, what I want to know is which cells at the bottom are the first Italian ancestors, and which cells near the top are the North American Italian immigrants. And as we'll see, building family trees of both people and cells gets a lot harder to do after everyone has dispersed all over the place. So the way that this works during human brain development, the cellular dispersion of cells, if we look at the brain in cross-section as it's growing, we can see that we have the fluid-filled ventricle in the center of the brain, surrounded by the cerebral cortex. And now if we zoom into the cortex here, we can see the ventricular zone, or this inner surface closest to the ventricle, where neural stem cells are dividing and multiplying. Some of these cells grow along so-called radial processes that reach out to the outer surface of the developing brain. Then as development proceeds, cells start to divide in such a way that each doll migrates up its mother cell's radial process away from the ventricle. The problem here is how to tell while this process is ongoing. So where is my great-grandmother Italian cell, and where are the immigrant children going? The answer to that question is by introducing DNA barcode labels into each mother cell that allows me to make the cells glow with a different random fluorescent color than the cells around them, and importantly, that the same barcoded color is inherited by all of the daughter cells going forward. And when you do this for a whole bunch of cells, then this is the result. So if we zoom in here to look at the ventricular surface where all of the mother stem cells are located, there are far too many colors for me to be able to tell the difference between all the different families of differently colored cells. But if we look up into the cortical plate up at the top, which is forming the cerebral cortex where all the daughter cells have migrated into, we can see that there's many different colors intermingling with one another in a way that's encouraging for this technique and has inspired ongoing work that will try to label fewer cells in order to be able to match them back to their individual lineages. And so the last thing that I wanted to tell you about this image is that it actually started with my advisor encouraging me to just give these color barcodes a try and see if they would work for lineage tracing. Worst case, he said, all you get out of it was a pretty picture. And I think that so far that's worked out pretty well. So in addition to giving me good advice on new techniques to try, I'm very lucky to have a wonderful mentor at Boston Children's Hospital, Chris Walsh, who works at this very important intersection of developmental biology and human clinical research. And this is made possible and enriched by the training I've gotten through another unique lineage, the union of Harvard Medical School and MIT, which gave rise to the Joint Program in Health Science and Technology, where I'm currently a PhD candidate. So also because of this, I've had the benefit of learning from and collaborating with people across many different labs, centers, and institutions, who I thank for their contributions to this project, as well as our funding sources. And I'm really excited to see what we can use this technique to teach us about the brain going forward. So stay tuned, and thanks for listening.